epithelial tissue. I told you epithelial tissue was one of the major types of tissue, just generally. So I guess you could think of this as subtypes, specifically of the type epithelial tissue. Whatever is easiest for you to think about. Yes, there's only 4 major tissue types, but within each 4 major type, there's lots of subtypes. So we're going to start with epithelium. There's plenty of different types. And we classify the different types in 2 different ways. First of all, number of layers. So the basic epithelial tissue that I showed you in the previous videos, so it's just like a single layer, which means, yeah, just 1 layer of cells. Some epithelial tissue has just 1 layer like that, but there's others that have multiple layers. And I don't just mean like 3-dimensional sheet, because the dimensions go off here. I mean, I guess vertically, if you think of it that way, there's multiple layers. And I'm just going to draw free. That means the basement membrane is all the way down here at the bottom. So these are the only cells that touch the basement membrane, and the cells at the top are the ones whose apical surface actually is in contact with the world. So simple means you only have 1 layer. Stratified means greater than 1 layer. Okay, then you have something called pseudostratified. We'll look at that later when we actually get to that specific tissue. I'll just tell you, pseudo means like fake, not the real thing. This particular tissue looks like it has multiple layers, but it doesn't. That'll make more sense once we actually get to it. I assure you, we will get to it. The other way that you can classify epithelial tissue is by the shape of the cells. Squamous cells are flat. In fact, that word squamous just simply means flat. So a squamous cell, those aren't squamous. Squamous cell would be like this, kind of flat. I'm sticking a nucleus in each one. Compared that with a cuboidal cell. So here's our squamous cell. And then our cuboidal cell is shaped like a cube. So if I could draw in 3 dimensions, it's, kind of squarish when you look at it on a slide. But in 3 dimensions, it would be a cube. And then, so that's cuboidal. And then columnar. Columnar are really tall and skinny cells like this. That's a columnar cell. They're very tall, very skinny. And actually the distinction between cuboidal and columnar is actually pretty straightforward. Columnar cells look really tall and skinny. Cuboidal look much chunkier, like a square. Okay, so what we'll do now is this somewhat painful process of just going slide by slide by slide and looking at all these different types. But before we do that, let's just talk generally about these microscope slides, because I'm willing to bet, I would not be surprised at all if all of you are seeing just, oh my God, it's a bunch of pink and purple dots and some lines, some squigglies, and how am I ever going to tell all these apart? That is the initial reaction to epithelial tissue, or really just tissues in general. And I feel you, they all kind of initially look the same. The best way to learn this or any other anatomical part is you're going to look at it over and over again. And you're going to start trying to really pick out some features that'll give you some information about what kind of tissue am I looking at. Because a major part of your exam in both lecture and lab is going to be identify what type of tissue this is. So first of all, I'll just tell you, why is it always pink and purple? Often, not always. That's because of the dyes that are used when making these when making these slide preparations. So if we just put in the tissue without any kind of dye, or I'm getting emails occasionally. Sorry, I should mute I don't know if I mute my computer, if that's going to also mute the audio. So I'm just going to leave it, and we're just going to listen to my emails come in now and then. Sorry. Okay, so if you just took, a chunk of an organ, and you prepared this thin slide, and you just stuck it under the microscope, you would see nothing, because this tissue is basically clear in appearance under the microscope. So what you have to do is add these particular stains that will bind to different parts of the cell and the other structures like the basement membrane. And so those cells happen to be often shades of purple and pink. Occasionally you'll see a bluish. All right, other things I want to note besides why they're those colors. These are all images from your book, from this table. So convenient because they gave you nice pictures of each type. Notice that they'll give you the type in the, a photograph of an actual tissue slide on the top here. So that's a photo. This is an artist's drawing. Now, the artist drawings can be really helpful when you're first getting started because the artist kind of highlights certain features that you need to draw your attention to, like this blue line here. That's the basement membrane. So the artist has kind of brought those features out for you. When you look here in the photograph, it's a little harder to see that basement membrane, but not bad. There's some other good examples of, in the drawing here, they tried to really highlight like this white thing here, which we'll talk about what that is later. Less easy to spot in the photograph. So when you're first learning these, it's kind of helpful to look down here at the drawings that have been provided for you. That one looks very, very similar. Some of these that one does too. But some of these, you can see a fairly, a fairly big difference between the artist who's really brought out the features and the actual photograph, where those features might be a little bit fuzzier. You can use these, I think, initially to help guide your eye towards certain features, but on the exam, this is what you're going to be tested on. On my exams, you're going to have photographs from these tables that I'm showing you in these slides, and that's what you're going to have to identify. Not these drawings, so just be warned. Okay. And we will practice identifying these, don't worry. I'm going to fly through all of them. Probably take me a couple videos to do that because I don't want to make it too painful for you all at once. So, again, I'm going to go through them all once, and then we're going to practice them several times in class. Oh, a couple other things to know. In addition to being able to identify the tissue in a given photo, I'm actually going to skip here. What else do you need to know? Because I always get asked this. And it's a fair question. What else do I need to know? Besides identifying this is actually the simple cuboidal epithelium. What else do I need to know? You need to know broadly what's the function. And some tissues, sometimes there's two or more tissues that have a particular function. So there's some functions in common across these tissues. The other thing is I'd like you to give me example of where to find it in the body. Sometimes I only expect one example. In some cases, a tissue is really common. Maybe you can come up with, let's say, one or two. Where do you get these examples? Well, they're all in your book. And also, I'm going to state them in the videos. I'm going to give you a couple examples. So don't worry about how on earth am I going to find an example. I'll give them to you, and your book has them as well. So you shouldn't have any trouble finding examples of all of these tissues that you should be able to tell me. If I ask you, give me an example of a place you'd find simple cuboidal epithelium. So you should have no problem at least getting that information. The trick is remembering it. And we'll work on that.